Welcome, everyone. Couple procedural notes. If I know some people try to do this on their phone, and maybe you already know by turning it sideways, you might get a better view. Also, the slide numbers. I hope you can see my cursor. The slide numbers are in the corner if you want to refer to them when we have our discussion section at the end. The decoration you see here is a cape on the Oregon coast on northwest North America, and it has provided a couple of nice collections so far. So what is an amateur? When I saw this comparison, I had to look it up, between amateurs and professionals, it confirmed for me that both can be experts. I certainly identify with the pastime aspect of this definition. Amateurs can wander, some even chaotically, often with little worry about funding. Loren Bahls, a professional diatomist turned amateur, likes to say that his work is supported by the U.S. Social Security Administration. The illustration on this page shows a tube-dwelling diatom from the Willamette River in Oregon. So today, ostensibly, we'll take a look at these purposes. But in a way, this talk is really about suggesting possibilities for amateurs and professionals alike. The decoration here is a diatom bloom on a coastal mudflat in the Netherlands that was provided by René van Wezel. So briefly, our topics look like this. I'll do the decoration first, though, because it's very distracting. It's a scanning electron micrograph of a valve of Ketonia elaborata from the Umaru diatomite in New Zealand. Many of you, maybe all of you know about this one. Pretty amazing. And so this is Bill Daly's one of our members of the Diatom Forum you'll hear about later, and Marianne Tiffany is a retired diatomist. Today we'll touch on a little history, some collaborations, a way to organize and search for images, and an online forum. Okay, a few amateurs in history. Amateurs are responsible for a great deal of the historic work and some of the continuing advances in diatom studies since diatoms were discovered by an amateur in England in the year 1703. Just three examples here. Friedrich Hustedt and Niels Foged were both high school teachers who began studying diatoms at a pastime. And Loren Bahls from the US, has made many contributions since his retirement. One of his works is this 2-volume atlas of diatom images. I'd just like to take a moment with this list of people that I've collaborated with over the last decade. They've helped me write collection reports, glossary terms. They've helped me enhance the online forum you will hear about and have helped me prepare this talk. Like diatoms, collaborators are all over the globe. So next, I'd like to look at an online collaboration. This is a species page from Diatoms of North America, otherwise known as DONA. These species are from the famous Pleistocene diatomite in this road cut in Terrebonne, Oregon. Some authors consider this to be an extinct species. Loren Bahls suggested that we pursue this possibility that this species might still be extant because the type location was from nearby fresh water. So began the chain. So I scraped off a little bit of it, probably right about there, and sent it to Raymond Hummelink in the Netherlands, who offered to clean it and make some slides. And the images you see here are from the slides he made. Loren Bahls again did most of the writing, And he also made sure it got into the Montana Diatom collection in Missoula, Montana. And finally, Sarah Spalding is our professional member of this collaboration. And she did the page editing, layout, and of course, supervised us so that we made it look like the rest of DONA. So let's see, what else I want to say about that. Okay, the numbering on the images here. This is a monoraphid species, meaning it has one raphe. And the number 1 is the raphe valve. You can see it here. And the number 2 is the rapheless valve. Here's the sternum of the valve without any raphe in it. A distinguishing feature of this species happens to be the greater number of areolae or holes between this hyaline area and the margin. The results that you see at the bottom here as part of our description on DONA points out it's another example of a scientific work that requires more work to figure out if it is truly a new species. So we, again, unresolved, but it was definitely worth a worthwhile effort. Our next topic delves into collaborations involving diatom glossaries. As many of you know, there's an extensive terminology used to describe the many aspects of diatom morphology and physiology. There are a few comprehensive glossaries available and many specialized glossaries, and we'll talk about a couple of these. Ross et al. From 1979 provides one of the most used glossaries. It is an extension of the earlier version from 1975. The 1975 version is probably the first comprehensive glossary written. And Barber and Haworth, their little booklet, I have found to be very useful for amateurs, professionals alike. It contains many line drawings of frustule morphology. Gogorev et al. From Russia is an extensive glossary, and it also contains some of the terminology that they use for the endemism that's in Lake Baikal. And so, of course, being such a massive region to cover, you can see that they would have to deal with that. I thank Mark Edlund for sending me this paper. And finally, the professional website, DONA, provides recent definitions, often with illustrations. A couple of specialized glossaries, and of course there's many out there. One by Eileen Cox talks about the coverings of the pores that we mentioned in the last slide. And Irina Kazmarska talks about the life cycle, and we'll come back to her a little later. So we'll emphasize the glossary on diatoms in North America, where I have been collaborating. Currently, it contains about 184 terms, and we think of it as a teaching tool. A lot of definitions that have leads to others in the glossary. Frustule, for example, we start out with that one, lists 6 other terms to explore. Since the first reported diatoms 320 years ago, many people have proposed terms for features visible in the microscope. As a result, agreeing on names and definitions can be challenging. We'll look at 3 of these challenges. Alveolus is a good example of one where, after working on it, we ended up providing only a simple basic definition, but there were many variations. After some efforts to find consensus, we decided that consulting the author of the group of interest might be the best way. At least this could provide some consistency within that group. We work on this glossary constantly, so we encourage you to check it for current uses of a term and to suggest updates and corrections. So here's an interesting one. Annulus means ring in Latin. And in this case here, here's the annulus. And in this case, it is an unornamented ring shown here. Take a look at the complete definition on DONA for a fuller explanation and references and so on, more photographs. And then here's an interesting variation. So this is a homonym in the glossary. Annulus can mean a ring or a collection of specialized striae. Quite a difference. As you see by the description, the term was based on a structure that is visible in a particular species. Now it's called Geisleria, and previously it was called Navicula annulata. And so the annulata, as you may be guessing, it was this ring structure. Much later, Gogorev et al. Developed a different term, which that glossary uses, and that one avoids this problem. Again, take a look at DONA for a more complete definition and more photographs. See if we want to say anything else about that one. No, I guess we're good on that. Okay. So this was not a collaboration really, but I wanted to highlight this one because I felt it was very important. Here's an example of a definition that we're using to draw attention to an expansive specialized glossary that is also a large collaboration, professional collaboration, albeit among professionals, with authors from Canada, Japan, Minnesota, and Great Britain. Again, I thank Mark Edlin of the St. Croix Watershed Research Station for telling me about this paper. This definition written here is the barest of introductions, as you can see. And down here at the bottom is an example of the pointers we provide when we can to other terms that are in DONA so that people can move around and maybe learn some more things about the terms. Okay, moving on to another area of endeavor, amateur endeavor for sure. So this is a method I use to organize my images. I'm thinking that it might generate some discussion at the end. Over the last 13 years, I have accumulated about 10,000 images. About 4,000 of these I have edited so they are presentable. This 4-step scheme takes a fair amount of work to set up, but it helps me find things quickly and lets me organize them in various ways. Let's walk through the process. Metadata, as you know, is metadata about data. In this case, EXIF is the metadata, and it means exchangeable image format. This metadata is actually stored with the images. If you copy or send an image in its original form, all the EXIF data will go with it. There are pros and cons to this in terms of size of image and privacy. I use Microsoft products here, but I imagine that other operating systems such as Apple may have similar ways to work with EXIF metadata. So we start by adding metadata to the images. I've become fairly bold with the amount of information I include in the tags. That's how it shows up, and the comments, and have not found limits yet. You can put in as many tags as you want in any order. This disordered list of tags is kind of humorous in a way, can then be used to find images, as you'll see. In my collection of 10,000 images, the searching is very fast. I have adopted hierarchies as my scheme to help me sort out things. So these are locations, collections, names, and so on and so forth. Comments can also be quite long. I try to keep references in these comments so that I can store the references with citations with the image. So in the next step, we have to get all that gobbledygook out and put it and eventually get it into the database. I keep the export process in a script called a batch file so that I don't have to rewrite it every time. Here is the line of code that does the exporting. The program exif tool is used, actually does the exporting, and it ends up in a CSV file, and that, that's the code for the CSV file. So in the next step, we import the metadata. I use this series of buttons to help automate the process. The second button turns the mass of tags into a list that can be sorted and filtered. So turns the gobbledygook into this. So when you look at it in the database, this is what you would get. And of course, you can see now, once you got it like this, you can do, in the database, you can do almost anything you want with it. Finally, we use the data to write some reports. And the data can also be exported to use as captions in more customized reports in word processors. So here you can see that's the name that was in the metadata, and I used a query to capitalize it to look better. And down here is, so this is a part of a tag, or not the number, but this part here. And this is the comment from the metadata. Okay. So if the database busyness is too much, you can stop at the first step and just use EXIF data to search and organize your images. Here's the complete tag list that you saw before. And here's the search string I used. And as you can see, I only need, when we finally, okay, here is the search, the place where you place the search bar. And you can see all I needed for this search was 4 words. And that's all I need. And of course, it implies that I pretty much know my data. So that is one thing. This search of 10,000 images was almost immediate to return these 2 records. One of the results, one of the result rows here is in blue with its image in the details pane. And you can see the tags are down in here and the comments are down in here. You can write more complex search strings. I have found the syntax worth learning through the documentation, though the documentation is a bit scattered. Comments, authors, dates, and so on can also be searched. Okay, so moving on to another lively amateur project. Here's the email group Diatom Forum. This might be a good time to take a breath and have a drink of water. This is the home page of this email group, as seen if you search the web for Diatom Forum. So here's a place you can sign up. Here's a place you log in if you're a member. And if you wanted to go back and look at our homepage, you can see that. And here are the messages and topics. And over here, you see something called hashtags, which you can use to get groups. And here are the hashtags here, and you can look up things. Well, I want to know about mounting. So you can go to hashtags and look up mounting, something like that. The public can't own a search bar up here. This is very useful. The public can read messages and wikis and download message attachments. Professionals as well as amateurs may find that searching the messages provides all the information they need. However, if you become a member, you get to do more things. So we have a place where you can write messages. Actually, if you join, you'll quickly learn that you can use whatever client you use locally. You don't have to come in here and do it, although being in here lets you do other things. Let's see. We're going to talk about, again, you see your hashtags. There's a wiki at the bottom. We're just beginning to play with databases. It's not the most, how to say, it's a little laborious to use, I think, but might be useful. Files, we have a lot of files stored in here of all kinds of diatomaceous material. Our members include people working and retired from many walks of life, and amateur microscopists with expertise in photography and microscope techniques. Currently, we have about 160 members. And that may be shocking in a little way when you think about some of the groups that have tens of thousands of members. Even with this minuscule membership, it is a very enthusiastic group. The 4 topics that appear here give you a sense of the range of interests in the forum. So we talk about old microscopes sometimes. And here's one on, adding more photographs to it. That's a notice. And here's somebody working on older diatomist, older amateur material. And here's a group of people who are working with UV illumination. And they get, of course, you get some really striking images. So I encourage you to join. You can even control how much email you receive, down to none at all. Okay, so here's an example. If you're a member, you can see all the work that people have posted in their albums. And you can see that there's quite a range, again, of things that have been done and things that people are interested in. And you can see how chaos can ensue with these albums, but I guess that's part of the fun. One of the hot topics on the forum is creating slides of diatom arrangements. Members share some very involved techniques for preparing material and for making the slides. You saw this arrangement on the forum homepage. Some of our members are continuing this 19th century tradition while providing many innovations. So how do they do these arrangements? Well, here's a very brief demonstration. You can imagine that making an arrangement is a painstaking process. Here's a demonstration of mounting a diatom valve by Stephen Nodge. He learned this technique from the late Klaus Kemp. The sample was collected by forum member Bill Daly in 2024 from a Cretaceous deposit in California that was laid down before the extinction of the dinosaurs. So yes, dinosaurs survived the Cretaceous meteor impact. So just going through this a little bit, in this needle step where we actually are going to pick up a diatom for placing on the arrangement, in this case, Stephen is using a glass needle. The needle itself starts from about here to the tip, and this is a chopped off hypodermic needle that's been, and the glass has been placed inside it and glued by a little interesting little technique. I've also heard of a diatomist who currently uses a hedgehog guard hair on a wooden handle. I think that would be really interesting to try. So here you see he's picked up a valve, and here you see magnified, and this valve is about 40 microns in diameter. And here's an example of a different diatom, but a mount. And this one's been photographed as a stacked image of Gloriopticus. Pretty great name. Again, this happens to also be a fossil diatom. And now here's a small gallery of arrangements by Diatom Forum members. This one was arranged and photographed by Stephen Nadge, and he got the special effects, of course, with the kind of illumination that he used. This one was arranged by the late Klaus Kemp, and this one has a 106 diatoms in it. Give you a chance to take a look at that one. You probably know all the genera for this. And finally, we have a 247 form slide. The slide, of course, is over here. Here's where the arrangement is. So this Triskele was done by Stephen Beats from the UK. So join the forum so you can see more of these images. To finish, here's one of my favorite scenes of living diatoms. I saw this cell of Cimbella moving along to the left, dragging this single cell of Melisira. Thank you, everyone, for this chance to talk about diatoms. Here are a couple of discussion questions. There's actually a definition in the glossary right now that begs this question. And so, that might be one we could ask. And I'd like to also hear about methods that you folks use to organize your images. Thank you.
Hey, Rob, can you go ahead and turn your camera back on for us?
Okay, let's see. Where do I go? Oh, there it is.
Down at the bottom video.
Got it.
Oh, good. Thank you. Thanks so much for that conversation. Before we get into these discussion questions, you can leave that up there. I like the questions right there.
Sure.
Well, tell us a little bit. Tell us a little bit about how you got into this. Like, what did you do before you retired, and that you landed on a love for microscopy and diatoms?
Well, as often happens, it started with a microscope in 7th grade, a decent quality Bosch and Lomb microscope, from the turn of the last century. And then in college, I was actually introduced to diatoms by a professor. And so I went on to study botany, and most of my work has been in vascular botany as a teacher and so on, then computer programming, and then when I retired, I fussed with various things, and then suddenly I realized that diatoms were still out there. So I guess in about 2010, I got the microscope that I have, and I started working it in earnest. Quickly discovered diatoms of the United States, now diatoms of North America, and started offering little edits to Sarah, and then she invited me to work on the glossary.
Nice.
So there we are.
That's great. That's great. I want everyone to go down and type in the message how old they were when they first learned about diatoms. I'm gonna write mine. I'm gonna write mine.
Okay.
I was a sophomore in college. There you go. I'm curious how, what are we missing here? Not getting our kids to see diatoms.
I know. I was not a pushy parent, and my kids just decided that's fine, yeah. And they've had comments about my hobby, so
yeah.
That
college senior.
That's right.
Senior, look at this, senior in college. Yeah, see, what in the world? We're all studying, we're all getting together to learn about diatoms, and we didn't learn about them until we were in our 20s almost. So crazy. Rob, tell me a little bit about I want to get into your image organization. What kind of scope and what kind of camera system are you using? Could you explain that to us, please?
Sure. I have a Nikon LaboPhoto 2. I've got pretty basic objectives. It's all bright field.
Mm-hmm.
I have a 1.4 NA pair of condenser and objective. I use those for Well, this image here is a 40x
Mhm.
objective. The camera is a consumer grade Canon and mounted on the top.
A DSL or what kind?
Yes, DSL.
Oh, okay.
Yes, and it's worked very well for me. Yep. So I don't know what else it can. I have a very sophisticated method that I use for oblique illumination.
Mm-hmm.
I have a 3x5 card that I cut across, and I lay it halfway across the field condenser, and I can get some sometimes surprising images.
The rest of us all learned by sticking our finger under there to do it.
There you go. That's the first technique my professor taught me about. Yeah.
I remember the first time I saw that too. Um, anyway, let's get to your, let me see if we have any questions coming up in here. I love that. I mean, look at this. Third year college, senior, master's, freshman in college, second year, 20 years old. It's just like, what the heck? I mean, it doesn't take that great of a microscope to see diatoms.
No,
We got to make sure our kids are starting to see these things,
I know. We
especially
have,
as important as they are.
right.
Yep. Um, let's talk a little bit about your first question here. Can an online resource like the glossary be used as a primary reference?
Yeah.
Um, I would say for sure it can be.
Okay.
I think I would. How would I do it? I want to like get on the backside of my computer here and start looking
Mhm.
at DONA real quick and
Well, take a look at the definition for Christa marginalis.
yeah.
That's the one that to me jumped out as an effort to combine the information to the point where we could
Mm-hmm.
use DONA as a reference.
Sure, sure. We have, I think if I was writing a paper, I think what I would do in that situation is just say, um, I would just say, valve terminology or terminology follows Diatoms of North America and
Mhm.
cite our sort of our standard citation that we've got now, the
Mhm.
2021 citation. I think that
Mhm.
would be a starter, but it's interesting that we haven't actually, like for species pages, we actually can give credit to whoever communicated that thing.
Mhm.
Whereas
Mhm.
with glossary, we haven't done that. And I do want to also say, um, I also do want to say that extend my major thanks to you, Rob, for helping
Oh,
out with this glossary.
well, it's certainly a great project.
Yeah, yeah.
I really enjoy it.
Um, I'm gonna quick. As I see Matt Ashworth has got his hand raised. Matt, go ahead and unmute yourself. You can turn on your camera. There isn't that many of us today. We can do that and ask a question, please. Hey, Matt.
Hey, Matt.
Hey guys, just wanted to comment on the citation of the glossary, but Mark sort of answered the question for me that Rob has actually been very communicative and very helpful as we've attempted to bring some order to the chaos of diatom taxonomy and terminology. And yeah, it's just been a matter of a statement in the material and methods about, the terminology and what we're using. And yeah, just citing the Spalding et al. Citation at the bottom for the web page and web project as a whole. But
Mhm.
yeah, please, everyone, please cite where you're getting your terminology from, because
Right.
yeah, it is very difficult to find a common a common set of terminology,
Yeah.
and the work Rob has been doing to try to collate all of this information together has been really helpful. So
Great. I'm glad you like it. Do you think um, when not that I'm writing papers, but do you think when you cite it, is the citation, well, Mark said the general citation, is that enough, or do we need citations for the actual page that the term is on? What would be appropriate?
Well, I mean, I think the glossary is fairly obvious when you get to the when you
Yeah.
get to the homepage.
Yeah.
So, I don't know. I don't think people have a problem finding it
Great.
if
Great.
if they need to get to it in terms of citing so that people's hard work is properly credited. I don't know. I don't know what else we can do about that. But
Yeah, that probably is definitely the simplest. Yeah. Thank you.
Yeah, I think I want to.
thank you.
Yeah, I want to follow up and just say to everyone, if you see a term that either could use a little tweaking or a term that isn't in the glossary on DONA, please reach out to us, reach out to Rob.
Yes.
Help, provide help Rob, give Rob, a direction, a reference, anything that, can help pen that, or images even that can help pen that, that glossary.
Yeah. Images are welcome. You can find my email address on my little page that's about in the About section on DONA.
Well, I was also intrigued with your very formal way to deal with images.
Okay.
And I think you probably have a lot of people listening that are envious of you, because I don't know how many of us are doing that
Well,
at that level.
I'm happy to share the work of the last 10 years.
Yeah.
And to be honest, the business of tagging stuff, as you could probably guess by that I'll go back to those pages if you want me to,
Yeah,
but
go for it.
you want to do that? Let's see.
Yeah.
How do I?
Well, you just click backwards if you want.
Yeah, I'll
Yep.
go slowly so you can enjoy the pictures again.
There you go.
All right, so
Oh,
I'll back up to, well, I'll go
yeah,
back to,
right here.
okay. So here's here's the mess, right? And so you can see, you got a lot of decisions to make about your tags. And yeah, I end up curating the tags, from time to time, make sure I haven't duplicated and stuff like that. What to say? So yeah, that's the first part.
So
Mark,
are you adding these, when are you adding these tags? So the image comes off of the camera
right?
and it has this EXIF data attached to it?
It does.
It
And
does.
so if you're, I apologize to Apple users. I don't know Apple. So I'm just going to talk about Windows here. So if
Okay.
you're in Windows, well, let me go show you more what the Windows screen actually looks like. So here's what you see.
Mm-hmm.
And if you turn on the details, it's hidden on my screen. It's hidden under if you move the pictures over, how do I move the pictures over? Oh, there you go.
Oh.
All right. So here's a thing. There's a tag, and I'm on Windows 11. So here's a tag under details. You turn it on, you get all this stuff. That's all EXIF data.
Hmm.
So as you may know, every time you take a picture, it stores all this information. Well, not the tags and the comments. You get to do that.
Yep.
But there's way more stuff than anybody, I think, would ever want to know about that photograph. So what I do, I take a picture, I load it up on the computer, and then I go through them and I start tagging them. And this thing here, I can actually show you, I guess, if you want me to, but I can go back and show you how to do it. But I don't know if that's something we want to do here. But there's this pretty simple way. I can do this offline too if people have questions. A simple way I'm supposed to use my tag. Okay, so you go into the tags,
Yep.
and there's a tiny little editor in there. Well, the editor is a bit of a pain, but it lets you put things in, and it has some sophistication to it. So, but anyway, I often do my tags as a group in a text editor like Notepad, and then I paste them right in here.
Mm-hmm.
So that works. So that's the tags. And then with the comments, like I said, I put a lot of comments in here. I put references in here. I put techniques in here. I put, how big my scale is. I just go for it. Okay. So that's,
That's, go ahead.
Sorry. So Rob, do you mind telling us is the EXIF, is it like a part of your camera system, or is it like a separate tool that you installed?
the EXIF data is part of your camera. And this is an international standard. So, they worked out a method so that it's everybody can use it. And so, like I mentioned, I think I mentioned, when you copy a full size image without modifying it too much and stuff like that, all of this information goes with that image. It's all there. And so, that can be helpful, or it has some cons to it too. Did that answer your question?
Just to follow up, so what kind of software do you use for capturing your images? I think you mentioned that before.
Oh, right. I actually use, let's see. Well, of course, the cameras, yeah, maybe that was your question. The camera's capturing the image, right?
Yep.
Did that make sense? And then I transfer it from the camera to the computer. I actually use something, are you talking about Photoshop and that kind of thing, or?
No, even before that.
Okay. So yeah, my workflow is basically take the picture, transfer the image to a holding folder, and then I go into what you're looking at right here.
Mm-hmm.
I go into this, which is it's Windows Explorer. And then I add my tags and comments. I try not to forget to do it. And then I run it through my process so that I can play with it in a database. So this is what you're looking at here, is the way that I get the tags in there. And it's part of the operating system of Windows. And so, other software you see, like Photoshop has a way you can tag things, but I don't use Photoshop. I have a sort of a simpler version of it, which I really, really like, and it's way cheap. How to say, well, with Photoshop, I'm not sure that it goes into a database that you can use separately from Photoshop. With Windows, it's on your machine. The database containing all these tags is on your machine, besides being in the photograph. I mean, the main source is the photograph, which is good. How to say? Yeah, and so if I give you this photograph, you'd open it up and you'd see all the tags and the comments.
Mm-hmm.
And then actually Windows will actually probably start capturing the tags that are in there and put it locally.
Great, thank you.
Sure.
Yeah, this is really impressive.
Well, what I've thought about, I mean, there's a little bit of programming that I did, because I didn't mention that between, Diatoms as a kid and Diatoms as a grown-up, I did database programming, so it helped. And I'm willing to share code or whatever if people want to pursue it a little bit.
Yeah.
And the batch file that I use for the download, I'm certain that that to me is definitely share that.
Yeah, I think that would be interesting. So just out of curiosity, Anna's asked, how do you make yourself go back and tag every single photo. And I think I just want to put it so you're even going back and adding scale bars and stuff to the photos, right?
Yes, and you know, here's an example right here.
Yeah, yeah. So that your camera system isn't even putting a scale bar on there for you.
No, you know, I've got a simple setup here.
Oh, that's okay. That's cool.
I wish I had fancier software, but I don't.
No, I get it.
Yeah.
That's fine. Oh, this is
Yeah.
interesting. I
Wait.
came from the old days when, you know, we did this with, you know, photographic negatives and stuff. And
Right.
you know,
Right.
we just put everything in a little
Oh,
3-ring binder with our negatives and our Kodachromes and our our prints if we had them. And
that
everything was
must,
very well organized for us. You just had to go back and look through them. But this, I mean,
right.
nowadays, in the amount of time that it used to take, you know, to shoot and develop and print a 24 roll of film, you can take 1,000 images
Yeah.
if you want.
Yeah, I know. It's
Digital.
bizarre.
So,
Anna asked, I think, another part to the question. What did she say again?
oh, how do you make yourself tag every single photo?
This is so boring. Doing this is so boring. And I have to force myself. I really do have to. I'm not a repetitive kind of, I don't do well with repetitive tasks. So, you know, I take lots of breaks and I just come back to it. And, you know, it's probably good for me and a little bit of discipline and focuses me on setting up these tags. And as you saw, you know, there's some value. Well, I feel here's the tag list when it comes out
Yep.
of the database. And to me, there's value in some organization. And you know, you can see there's lots of, I love hierarchy. So here's a hierarchy I try to use, and I separate the levels with periods. And but yeah, I mean, the payback, as you saw, is that I can pull up out of this crazy, well, I'm sure you folks have way more images than I have, but you know, out of my crazy list of images, the current the current operating systems are very fast, and these just come right back. So, you know, that's the payoff for the
Sure.
crazy work.
I'm impressed. I'm impressed.
Yeah.
All right. Any last questions, folks, as we're starting to head towards the top of the hour, right? You can oh, we got, hey, Layla, go ahead and unmute yourself and ask a question, please. Oh, come on, Layla. You got it. Do we have to?
I can see her view.
We can see her too.
Oh, yeah. Okay.
Uh-oh.
Did I do something?
No, you didn't do anything.
Right.
You didn't do anything.
Right.
So anyway. Layla, if you can't get yourself unmuted, go ahead and you can type it into our thing as well. Just maybe as kind of some final thoughts here, Rob. I mean, you've shown, you know, really clearly how you know, you as an amateur can really contribute to our broader diatom efforts especially in your work on the glossary. What other ways do you see? I mean, I think for some of us that there's a bunch of amateurs that are as jazzed up about diatoms as we are. You know, what are some other ways that we can think about working together in this world?
Right. Well, I mean, Klaus Kemp is an example here. He collaborated a lot in Britain. The name is skipping me at the moment. Paddock?
Yeah,
maybe?
yep. Kemp and
Okay.
Paddock. Yep.
Okay. He did a lot of collaboration, for instance, on Melisira. And he provided, I'm sure, maybe he provided slides and did other mounting kinds of things. So, you know, maybe the processing of slides or preparing of slides even. I mean,
Yeah.
you know, we have several living diatomists right now. Some were on the call maybe, who are really good at this.
Yeah.
And so that might be a way to do it. Some of the techniques that they have developed are quite something. With particularly with working with the chemicals around mounting, around cleaning. Yeah, I mean, we have a thread on the website about collecting, for instance, that's awesome.
Sure.
And cleaning. We have people on the site that are experts at cleaning. They do amazing work, a lot of hot acid work. But and so, you know, and they're trading information like
Yeah.
so on. So that might be, you know, techniques, and they might actually be interested in working on actual samples.
Oh, neat. Okay, that's cool. Layla did type her question in and just wanted to get your opinion on here. She asked about JPEGs versus TIFF files. Which is better, and what do you use? And you know, do you code your images as such, I guess?
Right. JPEGs are a huge standard.
Yeah.
And, you know, we all know that. I'm trying to go back here. And so they're such a huge standard that the operating systems on computers come already built in with software to handle them, you know, like the tags.
Yep.
And so I use almost exclusively JPEGs just because they're fast. I mean, when I look at the image quality, you know, this is off my microscope, but you know, the stuff we use on Dona is all JPEGs.
Sure.
And TIFFs, I mean, I don't know much about TIFFs. I guess you can get better image quality out of them. But from the work I've done, you know, just online here or on a screen, it looks like the quality is right there. It doesn't seem like people need more. And it's kind of surprising to me in a way that small images have so much quality in them, you
Yeah.
know. So
Okay. Thanks.
sure.
Well, that brings us to the end of the hour here. I want to thank Rob for joining us today and sharing some of the information. I think we may follow up a little bit with this image management works that you do, and really appreciate your introducing us to that thing.
Sure.
Remember, in 2 weeks, we do have David Hernandez Becerril coming back from Mexico, going to share some marine planktonic diatom information from the Mexican Pacific. And we did share some earlier links to either our efforts to fundraise a bit for Diatoms of North America, as well as a survey of how you are using diatoms.org, as well as how you would like to see it improved in any way, shape, or form. So appreciate your comments on that. If you've got friends, colleagues, or yourself who'd like to, you know, present on the Diatom Web Academy, it's a great way to reach out. Remember, all of these presentations are recorded and shared in a week or two on our YouTube channel. So thanks again to Rob for your presentation, and looking forward to seeing everyone in 2 weeks. Thanks, folks.
Bye-bye.
Bye.
Thanks, Rob.